always loved eating fruits and vegetables, and now I get to discover how our body uses our food to do a lot of cool things. Before attending the University of Wisconsin-Madison, I grew up in Manitowoc, Wisconsin, where I had a garden in my backyard, but I never really understood much about the food I was helping to grow. So I hope you'll use this experiment to investigate some of the science behind carrots from wherever you are. But before starting this activity, please get permission from a responsible adult.
So we're going to be doing an experiment using carrots to see how osmosis works in real life. So the first thing we're going to do is we're going to kind of go over how osmosis works. So in our body we have many, many cells, and plants have cells too. So the carrot we're going to be using is going to have cells. And in order to maintain balance, water can move into or out of cells following other things like salt or chlorine, any sort of molecule. So in some cases, water comes into a cell at the same rate it goes out. This would be in an isotonic solution. And so the cell stays the same. Now in other areas, more water goes into the cell because there might be more salt inside than goes out of the cell. This is called a hypotonic solution. In this case, the cell gets really big and almost stretched. So that's hypotonic. And then there's one more. In this case, the cell becomes shriveled as more water exits the cell than comes into the cell. This is when the cell is put in a hypertonic solution. So now using these basic concepts, we're going to explore this more using carrots and salt water. And I want you to think about how that might work and take a look at our carrots and see if you feel like they're stretched right now. Are they shrivelly? And then try and make a prediction of what you think is going to happen once we put them in salt water. So we're going to start by going over the materials you'll need to do this. You'll need some water, 4 containers, some type of carrots. They should all be around the same size, as well as some iodized salt, and then also a tablespoon that you'll use to measure the salt with. Okay, now we're going to start setting up the experiment. We're going to take the water and we're going to put equal amounts of it into each of our 4 containers. You want to make sure you have enough water in each container to cover the top of the carrots. It doesn't matter what kind of water you're using. Next, we're going to take our salt. And in the first container, we're not going to put any salt, but then in the second container, we're going to put 1 tablespoon of salt. This third container, we're going to put 2 tablespoons of salt. And in the last one, we're going to put 3 tablespoons of salt. We just want to see the differing effects in our experiment. And then you're going to make sure that it's all mixed up evenly so that the salt is evenly spread throughout the water. Next, we're going to take our equally sized carrots and place 1 into each of the containers. Right now, they're very firm and they don't bend very easily because they're full of water. So now that we're all set up for the experiment, we're going to let 4 hours pass before we come back and look at the carrots. Now I want you to think about what might happen. So you have salt in the water, as well as lots of water in the carrots. So based upon what we learned about osmosis, take a few seconds and try and figure out what might happen to these carrots after these 4 hours. Okay, I want you to start by making observations of each of the carrots before we show you what happens? Are they full of water? Are they shriveled up? Are they bending? Are they very firm? Take a few seconds to pause the video and write those down. And now we're going to take a look at the carrots. So in the first container we had the carrot with no salt in it. And in our experiment, that carrot didn't bend very much and it stayed pretty firm. The next one, with 1 tablespoon of salt, bent a little bit. Third one bent even more. And the fourth one bent even more than that, as well as being shriveled up. So now I want you to take a second and think about why that would be, knowing what we know about osmosis and what we talked about earlier. So hopefully, based upon your experiment, you were also able to see that the carrot that was in the highest salt concentration was able to bend more, as well as being slightly more shriveled up. So what happened here is that water exited out of the carrot into the salt water, so that there was the same amount of salt per amount of water outside of the carrot as there was inside the carrot. And since the salt can't move into the carrot through the cell membrane, the water going out was the only option. Therefore, the more salt that was in the water, the more water moved out of the carrot, which made the carrot easier to bend and more flimsy. We hope you enjoyed this experiment and were able to learn something from it. Again, this is Katie with the American Society for Biochemistry and Molecular Biology at UW-Madison, and we want you to know that you can chase your dreams no matter where you are and do science no matter where you are.